So next we will talk about how we can take individual amino acids and put them together to make peptides and therefore proteins. So we create what's called a peptide bond. That's a very particular type of bond, but it's also known as an amide functional group. So if you remember amides from the organic chemistry chapters, what we're essentially making is a covalent linkage between the two ends of an amino acid, the amino and the acid group coming together to make that peptide. So as you can see here, I've drawn just a single amino acid with our NH3, our COO minus, and a second one. And we draw them, this is what's called a head to tail formation of the peptide bond. This is the carbonyl or the carboxylate ion, and this is the amino ion. Those are bonded together to create the amide, that is the peptide bond between the two. In this process, what we essentially lose is we lose two hydrogens and an oxygen in the form of water. So water is lost, so you can think about this called a condensation reaction. So when you think about condensation, you're thinking water. They come together to create now, so nitrogens, the alpha carbons, here's that carbonyl, here's the other nitrogen, the other C alpha, and the carbonyl. So now we've made this bond between this carbon and this nitrogen with the loss of water to create, if you remember this functional group here, C double bond O to a nitrogen, that is called an amide linkage. This is also known as a peptide bond. So the peptide bond is a type of linkage that is an amide linkage that you might remember from organic chemistry. So if you take and you put two of these amino acids together, you get what's called a dipeptide, kind of like a disaccharide. If you put three together, it'd be a tripeptide, kind of like a triglyceride, triacylglyceride from lipids. So again, you get those different terms, those di, tri, tetra, etc. Now we have oligopeptides. If you hear people say oligo just means some amino acids. You may also hear the term polypeptides. So poly meaning many, like a polysaccharide, for example. That just means many amino acids are stuck together. Proteins sometimes can be termed polypeptides. So if you hear the term protein or you hear the term polypeptide, they sometimes can be used interchangeably. So you should be able to draw a dipeptide and show the amide linkage, the peptide bond between the amino acids to point that out. So if we take a look, now we can start talking about what this means for proteins. So when we take a look at the structure of, in this case, let's say our dipeptide here, we have basically two ends. And those ends, and like with a protein, we have the same thing. We have two ends, a beginning and an end. So the beginning is called, conveniently, the N-terminus. And it's called the N-terminus, guess what? Why? Because it has the nitrogen here. I bet you can probably already guess what this is called over here. This is called the C-terminus, because this end of a polypeptide, or this end of the protein, ends with a C double bond O, CO minus, if you're at neutral pH, right? So you can look, right, we have the C-terminus has the free carbonyl, or COO minus group. The N-terminus has the free amine. So, right, N-terminus, free amine, C-terminus, free carboxylic acid or carboxylate ion, okay? So the number of peptide bonds that you have will always be one less than the number of amino acids. So here is a dipeptide for two amino acids, we have one peptide bond. If you have three amino acids, you would have, therefore, two peptide bonds. If you have 8 amino acids, you would have 7 peptide bonds. So for example, if we have an oligopeptide that has 10 amino acids, 10 minus 1 therefore is 9. We would have 9 peptide bonds present in that oligopeptide. So if we take a look though, regardless of what you have in your oligopeptide, your backbone will always be the same, because remember, every single amino acid always has an NH3, an alpha carbon, and a carbonyl. N, C alpha, carbonyl. If I drew another to make this a tripeptide, N, C alpha, carbonyl. N, C alpha, carbonyl. Excuse me. So if we take a look, that backbone, that repeating pattern is always the same. So as you can see here in the slides, you have N, C alpha, carbon. And I just put carbon there instead of the carbonyl group, but it's not the C alpha, it's the C double bond O. And then N, C alpha, C. N, C alpha, carbonyl. So you can see the picture from your book here. Here's your N-terminus we have the C alpha, the carbonyl, N, C alpha, carbonyl, N, C alpha, carbonyl. So if you have to draw, let's say, a tetrapeptide, so 4 amino acids, therefore 3 peptide bonds, you can already just draw the backbone without knowing anything else. And then you just add the R groups here, for example, like your alanine and your lysine and your methionine, right? And your aspartic acid or aspartate. Okay, so again, the backbone that's on proteins and peptides is always the same. It's always, right, nitrogen to the C alpha to a carbonyl. Peptide bond, nitrogen, C alpha, carbonyl. Peptide bond, nitrogen, C alpha, carbonyl. Over and over and over again. It's always the thing, always the same. So the only thing that will change when you're dealing with different proteins or different peptides are the R groups. And the R groups are what give you a bunch of different characteristics that we're going to talk about when we talk about protein structure. So, biggest thing to remember from this chapter, that structure dictates function. You have the incorrect structure, you have the incorrect function. Okay? So, sometimes what you will see when we're talking about the structures of proteins, you may see folded or native. These are kind of interchangeable terms. These basically mean here's the functional structure of that protein. It's the one that can do the job that it needs. A non-functional protein can be referred to either as a denatured, so native, denatured, okay, or unfolded, folded, unfolded protein. So we can use those to basically help us to determine, hey, a native protein has the correct structure and therefore the correct function. But if we take away that structure or we denature the protein, then we have the incorrect function or incorrect structure. But you can also get what are called protein aggregation diseases. So some of you may have heard of things like Alzheimer's, Parkinson's disease, things like this. These are actually where proteins start in their native fold, but somehow what we call aggregate. So they no longer have the correct structure anymore. Okay, they might be partially unfolded, but they may be aggregated. We won't get too much into this, but if you're interested in learning more about that, please let me know. So let's take a look at our protein structure levels. Again, biochemists are not necessarily creative people, so there are 4 levels of protein structure. And so when we look at these 4 levels of protein structure, they are basically named exactly as you kind of would logically think that. So the levels of protein structure are just primary, secondary, tertiary, and quaternary. So the levels of these protein structures are exactly what they kind of say. The levels that you have, primary, secondary, tertiary, and quaternary. So primary, secondary, and tertiary, these are required for all proteins. Quaternary structure is optional, meaning some proteins have that, but it depends upon is that structure imperative to the overall protein's function. If it does not need quaternary structure to function appropriately, it doesn't necessarily need to have it. Okay? So the primary structure in protein structure is just the linear sequence of amino acids, similar to what we just saw in the last slide where you just have your amino acids and you draw the peptide bonds and you draw the R groups. But here's the thing. Somehow the protein knows exactly how to fold just with this. This is the only piece that's needed. These are the instructions to fold, but we don't necessarily have it all figured out of how they do that yet. But the linear sequence of amino acids, so alanine, glycine, methionine, etc., that is the primary structure. So you can just read those off, and that's what it is. Secondary structure, okay, our interactions are made with interactions of the backbones. This is why we talked about that backbone. Remember the backbone, when we look at a peptide or we look at a protein, it is basically, it just repeats, right? Remember, it's always N, C alpha, carbonyl. N, C alpha, carbonyl. Which means that because we have that, it's the only thing that repeats over and over again, we only actually have 2 types of secondary structure. Because the only interaction that we have, actually, this interaction, is a hydrogen bond. And it's hydrogen bonding between, there's a hydrogen here, this is a hydrogen bond donor, and a hydrogen bond acceptor on that carbonyl group. So we only have that as our interactions that we see in secondary structure. In tertiary structure, an easy way to remember that, this is the actual 3-dimensional or 3D structure of the protein when it is folded, okay? This is determined by interactions of the R groups. So again, this is why it's important to know which amino acids are polar, which ones are nonpolar, which ones are acidic or basic, because they may or may not interact with other amino acids or with water or with other species, with metals, for example, that have different charges, depending, okay? So again, there's only one type of interaction here, but with the R groups, we have polar, nonpolar, acidic, basic. There's a lot more variety in terms of what types of interactions we can have, which is why we get lots of different types of structures here. Finally, the quaternary structure are interactions of different polypeptide chains. So again, what this means, a polypeptide chain is basically a protein that has both an N-terminus and a C-terminus. So if you have, like let's say for example, we're going to talk about hemoglobin. Hemoglobin has 4 different polypeptide chains, and all 4 of those come together to make the overall structure that's required for the function for hemoglobin. So if you have quaternary structure, you might have, let's say, 3 different chains. So if you have 3 different chains, you're going to have 3 N-termini, and you will have 3 C-termini. Now, they might not all interact exactly the same, but we know that those 3 things have to come together, 3 different polypeptides coming together to create the entire protein. So these are the 4 levels of protein structure. So we're just going to kind of go a little bit more in depth using some of the information from your book. So if you take a look, here's just an example. This is actually insulin, okay? If you look, we have our A chain and our B chain, for example, but you can see here we have peptide bonds that will hold these amino acids together. We have glycine, isoleucine, valine, etc. as we go through, okay? So you have these are held together with those peptide bonds, remember those amide linkages that you have between the nitrogen and the carbon. So you have a covalent linkage between them. And again, you get an N-terminus and a C-terminus. So you can see in this example, skipping ahead to quaternary structure, we can see we have 1 N-terminus, 1 C-terminus here, 1 N-terminus here, and 1 C-terminus here. So we actually have 2 polypeptide chains. So this particular protein also has quaternary structure because of those interactions between those different chains. So if you take a look at the secondary structure, remember these are interactions of the backbone. So we have hydrogen bonding. Remember, hydrogen bonding is not a bond, it's an intermolecular force, which we will see again in chapter 17 because the double strands of DNA are held together with hydrogen bonding interactions. We have hydrogen bonds where we have the H is the hydrogen bond donor from the amine group of the backbone, and the carbonyl, C double bond O, is the hydrogen bond acceptor. Now they're not going to be directly right next to one another. You'll have to go a little ways difference so that you won't change the bond angles too much. But here's the nice thing, here's the easy way to remember secondary structure, right? There's 2 things that interact, okay? Donor, acceptor. 2 things that interact for secondary structure, which results in 2 types, right, of secondary structure. We have an alpha helix and a beta sheet. So the interactions of the backbone, there's only 2 species that are involved there, the hydrogen and the nitrogen and the carbonyl group. So because these are 2 groups, these are the only 2 groups that interact, that means we make 2 types of secondary structure, right, which is helpful that it's secondary. And these 2 types are what we call an alpha helix and a beta sheet. Okay, so we will look a little bit more in depth in those here in just a second. So if you take a look at an alpha helix, it looks like a helical kind of structure, similar to like the double helix that we have for DNA, or you can remember like a spiral staircase, these types of things. So as you can see in this example, remember the backbone has that regular repeating pattern. So alpha helices and beta sheets also have regular repeating structures and regular repeating patterns as a result. And because of this, so you can see in this illustration from your book, here is your alpha helix, and here's the hydrogen bond between the nitrogen, this is the hydrogen, and the carbonyl group. That hydrogen bonding is what holds the backbone together in an alpha helix. And the cool thing is about an alpha helix is that it is regular, and because we know the pattern of the backbone, an alpha helix will also always be the same all throughout. And what this leads to is it leads to what we call 3.6 amino acids per turn of an alpha helix. So if you start at one space and you go down just in one axis and you spin one full turn around an alpha helix, meaning 360 degrees, you have 3.6 amino acids. It's a very particular number, but we know that for sure because it gives us the information that tells us about the overall structure and how these 2 species interact to be a hydrogen bond donor and acceptor. With a beta sheet, though, we don't have exactly 3.6 amino acids per turn of a beta sheet. Beta sheets can come together from a large area from the beginning of a polypeptide to the end, so there's longer distances potentially. There's no exact number like we have for an alpha helix. But again, we still hold beta sheets together with hydrogen bonding, and it's still hydrogen bonding of the nitrogen from the backbone of the hydrogen and the carbonyl group from the carbon there. So here's just an example from your book of a bunch of beta sheets. So again, when we draw these nice cartoon-looking versions of an alpha helix or a beta sheet, you'll see alpha helices are kind of drawn like a helix, like a spiral staircase. But with beta sheets, we tend to use little arrows going either one way or another way. Okay, so if you see arrows like this, you know it's a beta sheet. If you see a little ribbon kind of diagram that has a little spiral staircase kind of feel to it, alpha helix. So you can see in this structure from your book, we have these beta sheets down here, and then we have these red alpha helices. Anything that's not in between is actually called a random coil. So it's not held together with hydrogen bond interactions at the backbone necessarily because it's not a regular structure. So that's what we look at when we're looking at secondary structure. So here's the thing about secondary structure. It's very regular. And when we have regular structures, right, when we have something that repeats over and over and over and over again, we just kind of get one type of secondary structure. So we talk about different types of proteins. There are two kind of general overviews. One is called the fibrous proteins. These are structural proteins. Structural proteins are proteins that are there to really help create stability in different things. So for example, your teeth and your bones have something called collagen in them. Maybe you've heard of collagen before, but collagen is a protein, and it's a very strong protein, right? So again, our bones, yes, are made of calcium and other things like this, but they use protein supports and collagen to help with that as well, which means you want them to be very, very strong. Okay, which means that they only have one type of secondary structure because that just makes them stronger if they just repeat over and over and over again to get that. Another example is something called alpha keratin or beta keratin, or maybe you just heard of keratin. Keratin's found in like your fingernails and your hair and things like this. So again, these are also proteins that are there for structural purposes, okay? You want your fingernails to be kind of strong, but they have a different texture than your hair, for example, or your type of hair, okay? So fibrous proteins tend to only have one type of secondary structure. Alpha keratin, for example, actually based on the name, it's alpha helical. Collagen, this is sort of a diagram, a little cartoon from your book here with the 3 helices. You just have 3 helices wrapped around one another, okay? If you get one type of secondary structure, you get that same pattern over and over and over again. It creates more stability, creates more integrity, things like this. But then finally we have globular proteins. So globular proteins, they don't necessarily just have one type of secondary structure, they can have many types. And that's because they have, again, a whole lot of different types of structures because they have a large diversity of function. They can be enzymes, they can be carrier proteins, they can be a whole bunch of things. Because remember, protein structure dictates function. For these fibrous proteins, their function is to create stability, right? To make sure our fingernails stay together, that our bones are strong, right? It's all about strong and structural integrity. So they have one type of secondary structure. For globular proteins, they might have to bind something and then let it go. Or they might have to bind something and then do chemistry and then make a product, like for enzymes, for example. So they might have a lot of different types of structures as a result. So if we take a look though, so now that segues into our tertiary structure. Remember, tertiary structure are interactions between the side chains. Now this is why knowing what those side chains are, whether they're polar, nonpolar, acidic, or basic is very important because the 3-dimensional structure of the protein, the backbone is, remember, all the same. We're not going to get a ton of different types of structures or a large diversity of structures as a result. There's 2 main types. But for tertiary structure, we have a lot of other types of interactions between those R groups. So here's just the figure from your book illustrating this a little bit better. But you can have something called a salt bridge. A salt bridge says, hey, I have a positive charge. You want to be near a negative charge. That's salt, because if you think about salts, salts have a cation and an anion. Same kind of thing here. We also have dispersion forces. These are basically where you put nonpolar amino acids near other nonpolar amino acids based on their size. So bigger amino acid R groups are going to have a little bit more space than smaller amino acids. Then we have disulfide bridge, so di meaning 2. These are 2 sulfurs coming together, okay? This is between the amino acid cysteine only. It's the only amino acid that can make a disulfide bridge. And then finally we can have hydrogen bonding. Also, again, not necessarily of the backbone, but of the R groups, because there are R groups that contain carbonyls, there are R groups that contain amino groups. And so you can have a hydrogen bond donor to an acceptor, or an OH group to an amino group, or an amino group to a carbonyl, an alcohol to a carbonyl, etc. So as you can see, with tertiary structure, we can get a lot more types of interactions. And because we can get a lot more types of interactions, we can get a large, more diverse subset of what the structures of these proteins are. So, however, not all proteins are just made up of amino acids. And this is why it's important where we talk about like vitamins or other cofactors or things. So cofactors are actually not amino acids, but they are requirements for a protein to do its job. So for example, maybe many of you have heard of hemoglobin. Hemoglobin is the protein that carries oxygen throughout our bodies. Well, hemoglobin is made up of amino acids because it's a protein, but the portion that actually interacts with oxygen to deliver it to different places is made up of iron. That's why sometimes when you donate blood, they will prick your finger and test, and they will say yes or no. They might say, oh, you can't donate today, your iron levels are too low. That's what they're measuring, is how much hemoglobin do you have in your system, and will you be okay to donate or give some away, for example. So if you look at the slides, you can see here's an example of myoglobin. You have an iron, right, surrounded by all these nitrogens. This heme group, what we call heme for hemoglobin, okay? This heme group, if you notice, is not amino acid based. There's a whole bunch of rings and nitrogens and irons, but again, so it's a non-amino acid portion of a protein, but that's required for the protein to do its job. Another example is maybe you've heard of the disease scurvy, or you may have heard of like people on boats for long periods of time would bring like lemons and limes with them because they needed vitamin C. Vitamin C is required for the protein that helps to make collagen. So functional collagen, I should say. So without getting too much into it, if you don't have vitamin C, it's not that you won't have collagen, you just won't have functional collagen. The enzyme, there's actually an enzyme called prolyl hydroxylase that requires vitamin C, and that actually goes on to collagen and makes it functional, essentially. And if you don't have vitamin C, you therefore don't have this functional enzyme, and if you don't have this functional enzyme, you don't make functional collagen, and then you get scurvy, and you can look up the Google pictures of what that looks like, or don't. Okay, so going back to our 4 levels of protein structure. So we've done primary, secondary, tertiary. We're now going to talk about quaternary. Quaternary is where you have 2 different polypeptide chains, meaning N-terminus to C-terminus, coming together. Now again, this is optional. It is required for proteins that need it to do their job. But if a protein does not need this to do its job, you don't have to. Hemoglobin, however, is an example that has all 4 levels. However, its cousin, myoglobin, just has the first 3 levels of protein structure. So the same types of interactions that you see coming together to make tertiary structure are the same ones that make quaternary structure. So for example, if we take a look at myoglobin, myoglobin only has 1 polypeptide chain. So there's myoglobin. It has 1 N-terminus, 1 C-terminus. It just has primary, secondary, and tertiary. But when you look at hemoglobin, hemoglobin has 4 polypeptide chains that come together. So I'm just going to draw them as little circles here. So you have 4 of them coming together. Each of them has their own N-terminus and C-terminus. So here you do have tertiary or quaternary structure as a result. And that's required for the proper function of hemoglobin as a result. So here just shows you overall an example using hemoglobin. So you can see the primary structure. Here's just the amino acid sequence. So you could do all 4 polypeptides, all their amino acid sequences would be the primary structure of hemoglobin. Secondary structure, there's a lot of alpha helices that are in hemoglobin. There is some turns and random coils, but we have a lot of alpha helices in that particular area. Remember, those are interactions of the backbone, the hydrogen bonding between the nitrogen-hydrogen and the carbonyl group. And then finally, we get the tertiary structure. So each of these 4 pieces of the quaternary structure, each of these has their own tertiary structure. But when they come together, then they create that quaternary structure. Again, within each of these polypeptides for hemoglobin, they each have their own heme group, the iron, right? That's not amino acid based, but is required for the protein to do its job. So there's an example of giving you the 4 levels of protein structure for hemoglobin, or the 3 levels of protein structure for myoglobin.